Yes, doctor.
Okay, now. All right, so this is the second unit for our lecture, right? So for lecture 2, when I talk about developmental genetics, okay, we also adopted a lot of these informations from Professor Jonathan's textbooks from Essential Genetic Biology, second edition and published by Blackwell Publishing Company in Australia. Right, so let's look at what is developmental mutants. Okay, so mutants that mean the organism that the genetics has been altered. Okay, whether it is knock in or knock out. Knock in means you knock the new gene inside the organism, or you overexpress it, or you knock it out. You took out that specific or particular gene and then you move it out. You knock it out. That's also called mutants. So of all of these genes in the genome, approximately 1 to 2% have functions that are specifically concerned with development. Okay, if we have 30,000 genes in the genome, yeah, so it's approximately 1 to 2%. So it's about 30 to 60 genes, but I believe this is too low. The number is too low that have functions that are specifically concerned with development. There are actually more, okay? And gene mutations carried by mutants organism may be spontaneous or induced by mutagenic treatment. You can induce with your mutation. There are so many mutations that we can use, chemical that we can use to induce mutagenesis, okay? And the different versions of the genes are called alleles. If you remember your STB2033, your general genetics, yeah, and there are usually just one normal allele that we call wild type. Still remember wild type?
Yes.
Okay, very good. So the totality of the nuclear DNA in an organism is known as the genome. Okay, and the specific combinations of the allele carried is what we call genotype. I hope you still remember this. And the totality of the characteristics of an organism is known as the phenotype, which is related to its visible appearance. So what you see, what you observe, that is what we call the phenotype. Remember?
Yes.
Okay, yes. Very good. Okay, good. So for the mutants, it is not possible to deduce the complete functions of a gene simply by looking at the mutants. If you look at the mutant phenotype, you cannot tell a lot. You can probably see the difference from the phenotype, but a lot of things that you still cannot see. So it is possible to deduce quite a lot about the normal gene's function by looking at the effect of the several mutants alleles at the same genes, but with different phenotypes. Okay, so we have the null mutation versus the allelic series. Null mutation means the gene has been totally knocked out, the whole genes. But if you look at certain allelic or partially knocked out gene, that's maybe different, yeah, from the total null mutant. Faham tak maksud dia? Ya, boleh faham tak?
Can you repeat again?
Okay, contohnya kalau kita tengok protein panjang kan, dia banyak domain, docking domain of the protein kan. Tidak ingat how protein look like? Contohnya satu gene, gene itu akan menjadi protein kan?
Yes.
Betul? Okay, dan protein ini dia mungkin berinteraksi dengan banyak protein yang lain, betul tak?
Yes.
Ya, so kalau null mutation maksudnya protein ini tidak boleh berinteraksi dengan protein yang lain. Ya? Boleh?
Boleh.
Right. Tetapi untuk partially knockout gene, dia mungkin ada certain domain yang masih boleh berinteraksi dengan protein yang lain. Tapi dia tak complete. Okay, that's why kalau kita tengok di sini, kita ada kelas A mutant, kelas B mutant, kelas C mutant, kelas D mutant dan wild type. Jadi kelas A, B, C, D ini dia menunjukkan phenotype yang berbeza, betul tak?
Yes.
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 So ini mungkin disebabkan oleh gene yang di knockout itu dia tidak complete. Jadi menyebabkan dia menunjukkan berbeza. Faham tak maksud tu?
Faham, doktor?
Ya, faham.
Boleh faham ah? Okay, good. So kalau kita tengok haplo sufficiency, saya harap slide ini sangat familiar untuk you all. So a weak loss of function mutant is more likely to be viable and so survive into adult life than a null. Faham tak maksud dia? Kalau null mutation mungkin dia adalah lethal, ya. Tetapi untuk a weak loss of function mutant dia masih boleh lagi hidup dan boleh survive ke dalam alam dewasa. Okay?
Okay, doktor.
Loss of function mutants are usually genetically recessive because the effect will be masked by a presence of a wild type allele on the other chromosome producing the normal gene products. So sometimes they are genetically dominant because loss of 50% of the products is sufficient to cause an abnormal phenotype. Contohnya kalau kita tengok incomplete dominance in horses. Kita ada criminal horses di mana kita ada palomino, kita ada light chestnut. Jadi ketiga-tiga ini menunjukkan apa? Phenotype yang berbeza tak?
Yes.
Betul.
Ya.
Yes.
Ya.
Yes.
Walaupun dia slightly loss of the function, tapi the genome yang dominant tu dia boleh cover it out dan memberikan phenotype yang berbeza. Ya. Dan juga kalau kita tengok snapdragon, bunga snapdragon ni, kalau kita tengok dia pun ada efek yang sama, betul tak?
Betul. Yes.
So you kena ingat STB2033 dia sangat-sangat penting. You sudah ambilkan untuk general genetics kan? And some of you, I taught you for that, betul?
Yes, doctor.
So this is very important. That's why kita general genetic you kena belajar dulu sebelum you datang ke tahun kedua. Dan saya pernah warning, beri amaran bahawa kalau you punya general genetic lemah, maka tak sampai ke second year nanti you akan menghadapi masalah yang susah. Apa masalahnya ialah you kalau tidak ada asas yang kukuh, you susah nak faham. Betul tak? Saya pernah tak warning macam ni?
Benar, doktor.
I attend my group. That's why saya kata you all really have to pay attention kepada yang asas tu. Okay? Kalau kita tengok gain of functions of mutant often express constitutively versions than normal genes product. So, kalau kita tengok dominant negative where the mutants form the genes product itself have no function but is interfere with the functions of the wild type form, which is from the dimer. So the recessivity of the mutants is generally indicated the loss of function, but the dominant may be due either to the haploinsufficiency or constitute all the dominant negative effect. So in principle, the dominant mutant types can be distinguished by the gene dosage analysis since the introducing the extra copy of the haploinsufficiency allele will have little effect, while the introducing the extra copy of the gain of functions allele will increase the effect. Yeah, so I hope you understand me based on the gene dosage. Okay, always remember if you introduce extra function of the haploinsufficiency have very little effect. Tapi kalau di extra copy effect yang lebih tinggi lagi. Kalau haploinsufficiency, walaupun dia increase satu copy, dia tipe jelas lagi.
Doctor, your mic.
Kita tengok X chromosome. In
Betul.
mammal, female has 2 X chromosome, while males only have 1 X and 1 Y. So in birth, it is the female chromosome types that the male have 2 Z, while two Z chromosome, we have the female having one W and one Z. In Drosophila, the sex is determined by the ratio of the X chromosome to the autosome, and the sex-linked mutations to the mammalians of X chromosome will be masked by the WhatsApp copy in female, which is the XX, but will produce a phenotype in male because the Y chromosome does not carry the corresponding locus. Kalau kita tengok di sini, X-linked recessive, the carrier of the mother. Kalau kita tengok kalau unaffected father dengan the carrier mother, ya, siapa yang akan mempunyai chance yang lebih tinggi untuk affected? Ialah lelaki, betul tak?
Betul.
Yes.
Okay, kerana lelaki akan inherit satu X daripada mak dia, betul tak?
Betul.
Yes.
Ya, dan dia lelaki dia memang ambil Y daripada bapak dia. Tapi oleh kerana yang X-link ini dia hanya link kepada kromosom X saja kan. So untuk perempuan dia tidak akan mendapat apa-apa efek. Tidak akan efek sama ada dia affected ataupun dia jadi carrier saja kerana X daripada bapak dia memang normal, betul tak?
Betul. Yes.
Kalau kita tengok perempuan di sini, anak perempuan dia dapat satu X yang normal daripada bapak, satu X normal daripada mak, jadi dia unaffected, betul tak? Untuk dapat tak sini? Nampak tak ini?
Yes, doktor.
Okay. Ya, kalau kita tengok yang anak kedua perempuan ini, dia walaupun dia dapat satu X yang affected daripada mak dia dan satu X yang tak affected daripada bapak dia, tapi dia hanya menjadi carrier. Dia tidak menunjukkan phenotype dia, betul tak?
Yes.
Ya, dan kalau lelaki yang mana dia dapat X yang normal daripada mak dia dan Y daripada bapak dia memang unaffected. Dia sempurna X ini. X yang dapat daripada mak dia sempurna, betul tak? Tidak ada kesan kan? Untuk yang ini.
Yes.
Jadi, tetapi untuk lelaki yang kedua di sini, oleh kerana dia dapat satu kopi daripada mak dia yang affected punya, dan daripada Y, memang dia, because ini yang bukan Y-link, dia X-link. So, lelaki ini memang affected. Kerana dia dapat Y dia, X dia daripada mak dia, betul tak?
Betul.
So, kita kena, ini, kalau X ni, biasanya lelaki akan lebih malang sikit lah kerana chances dia lebih tinggi kalau X-link. Yeah, because you don't get the X from the father, but you get the Y from the father. But you get the X from your mother. Okay?
Okay, doktor.
Okay.
Jelas tak ni?
Jelas.
Yes.
Jelas, doktor.
Okay, good. Okay, kalau kita tengok maternal and zygotic kan, okay, ini ialah egg, okay, the sperm. So the X dengan sperm dia akan menjadi zygote, dan lepas tu zygote dia akan jadi larvae. Okay, tapi kalau you tengok kan, yang X ni dia null mutation, negatif ni ialah null mutation, ya, boleh faham tak? Maksudnya telur tu, ovum itu adalah affected, ya. Okay. Dan lelaki ni contohnya normal because dia positif di sini. Tapi apa yang dihasilkan zygote nanti you tengok kepala dia hilang. Maksudnya abnormal kan larvae ini.
Ya.
Jadi ini maksud bahawa kalau kita tengok maternal dengan zygotic, maternal ini dia contribute kerana protein yang diperlukan ini dia tidak ada fungsi dia di dekat egg ni di sini. That's why apabila dilahirkan maka dia akan menjadi lethal kerana bahagian kepala dia memang interior dia memang hilang. Ya? Boleh faham tak?
Boleh.
Boleh, doktor.
Okay? Dan kalau kita tengok genetic pathway, if a set of genes are involved in a single pathway or process, is often possible to deduce the sequence in which they act from the genetic data. So there are two common situations. One where a group of different mutations have a similar phenotype, and the other where a group of mutations have two phenotypes that are in the same sense opposite to another. So where several genes have similar mutant phenotypes, the sequence of the action can sometimes be established by a rescue protocol. Kita boleh rescue dia. Contohnya di sini. Okay, double mutation phenotype. Okay, kita dapati di sini white, pigmented. Okay. So kalau kita tengok double mutants, kalau kita tengok A, you tahu tak yang atas ni palang di sini maksudnya dia adalah mutant. Okay, faham tak? Yang plus
Faham,
ini,
Doktor.
plus ni dia normal. Okay, so kalau kita tengok sini A dengan A dia tak ada lah di sini, memang square ni memang tak ada. Tapi kalau kita tengok mutant B dengan mutant bahagian A, dia akan menjadi white. And mutant A dan mutant C dia pigmented. Okay, and kalau B dengan B dia akan jadi di sini ya. So B dengan A, sorry, B dengan A di sini white. B dengan B dia memang tak ada lah di sini. Dan kalau C dengan B dia pigmented juga. Okay? Dan A dengan C pigmented. B and C also pigmented. So apa maksudnya di sini? What can you deduce from here? Ya, kalau kita tengok A, you tengok A dengan C ni kan dia pigmented, betul tak?
Yes.
B dia tak ada kan?
Yeah.
Yeah. So if you look at the combination, kadang-kadang kita boleh buat double knockout ataupun single knockout. It depend. Double knockout maksudnya kita knockout kedua-dua gene itu. Ya. Contohnya di sini. Okay. A with B. We knock out B, we knock out A, we get white. If we knock out A, we knock out C, we get pigmented. We knock out A and C pigmented. We knock out B and C also pigmented. Okay.
Okay.
So jadi kita boleh buat rescue. Rescue-nya maksudnya kita selamatkannya. Contohnya kalau kita knock out A, kita dapat mutants kan? Mutant A contohnya kan?
Yes, Doctor.
Contohnya we knock out A, we get the A mutant, right?
Yeah.
Yeah.
Okay, that means A punya gene sudah kita keluarkan, betul tak? We knock it out already kan?
Yeah.
And we see the phenotype. And now what we want to the rescue mean is that kita knock in the A gene sekarang. You try to rescue it. That means embryo ni sudah ada gene A yang kita mahu. So that is what we rescue. Kita knock in semula gene A itu. So we want to see the function. Do you understand my what I mean? Kita boleh knock out, kita boleh rescue dia juga. Maksudnya kita nak selamatkan dia. That is what you call rescue protocol.
Yeah, okay.
Yeah, we can knock out the gene. We also can do some rescue. We want to see whether kenapa kita buat rescue. We want to double confirm the function of the gene. We can do that. We double confirm it. Okay.
Yeah.
But that's why. Sorry. Don't be too happy if you have certain gene. Okay. The certain gene that you knock out, you look at the phenotype, you're so happy. Yeah, but you always need to double confirm the phenotype by doing some rescue experiment or protocol. Okay. So bila you publish paper, you kata, I knock it out, but the reviewer will ask, have you done any rescue experiment to confirm this phenotype? Yeah. So that's why kita perlukan, rescue protocol or a rescue experiment. Okay, boleh faham tak?
Boleh, Doktor.
Okay, good.
So
Okay.
Doctor, I have a question. So that means in practical experiment, So there's a possibility of getting different phenotypes before and after knocking the genes, is it?
Yes.
Like
Yes.
if I knock in, let's say I get red, and if I knock out and then I knock in again, I get blue, something like that. So is it possible to have these results?
It is possible because gene may act differently.
Okay.
Okay, you need to know that gene might not just play a role by itself. They may interact with other genes as well.
Oh, yeah.
Okay.
Yeah. Yeah.
You get this more you get this clearer when you study the coming lecture. Okay.
Okay. Thank you, doctor.
All right. So especially during the cell signaling ataupun gene communication lecture. So if you look at genetic mosaic, sometimes possible to make organism that consists of the mixture of the cell of different genotype. You see, a mixture of the different genotype. So there are genetic mosaic and can be useful as they provide information about where in the embryo in particular gene is required. So we know specifically the locations of the domain where the gene is expressed. Okay, so for instance, an embryo may consist of two territory, A and B, and BA particular mutant show a defect in B. Contohnya di sini, kalau kita tengok. Ya? Boleh? Kalau kita tengok ini teritori dia kan? Ini teritori. You tengok dia punya teritori boleh sampai sini. And this one dia boleh dikeliling dia juga. Can you see the how the genes is expressed in terms of the phenotype?
Yes, doctor.
Okay. Right. So if you look at genetic imprinting, this is also a very interesting punya experiment. So the nuclei transplanted from early blastomere to a enucleated egg. Enucleated eggs, that means the eggs, the nucleus of the egg already been, how to say, removed. Remember how Dolly was cloned?
Yes.
I show you that we remove the nucleus from the egg, right?
Yeah.
That one is called enucleated egg. Enucleated egg means the nucleus from the eggs already being removed. Okay, so can support developments, although the ability to do so fall off rapidly with stage. Okay, so the paternal-genetics diploid embryo developed poorly because of inadequate extra embryonic tissue, and the androgenic diploid have abundant extra embryonic tissue, but the embryo rests at the early stage. So this observation suggests that there is some differences between the state of the homologous genes on the chromosome from the two parents. Okay, so in genetic imprinting, some genes, okay, if you look at normal gene, they are biallelic expressed. That means one copy from the mother, one copy from the father, right? In a normal situation.
Yes.
Correct?
Yes.
Yeah, but for some special condition, a specific gene could only express from the father, but then silent from the mother. Even there are two copies, but one copy is silent. And vice versa, there is also gene that can only express from the mother, but silent from the father. Boleh faham maksud dia tak?
Yes.
Yes.
Yeah. So genetic imprinting also sangat-sangat interesting kalau kita tengok. Ya, contohnya Angelman syndrome, Prader-Willi syndrome, which is affect the chromosome 15, is because of the genetic imprinting. Okay, mungkin kita akan tengok in further further punya lecture, in the coming lecture nantilah. Yeah, you see. dekat sini. Kalau kita tengok di sini, ya, kita some of the gene can be being erased. Okay, contohnya di sini. Kalau kita di sini, kita tengok, ya, ini adalah germ cell, ini adalah sperm, and ini adalah the mother's genome. Kalau kita tengok di sini, ya, yang lelaki di sini, ini from the male kan, ini from the female kan.
Yes, doktor.
Ya,
Yes.
tapi kalau kita tengok di zygote ini, kita dapati dia memang ada yang daripada daripada ayah, juga ada pada mak kan.
Yes.
Ya, tapi ada segelintirnya akan di silent. Ya, sebahagian segelintirnya akan di silent, akan ditutup. Jadi dia tidak expressed. Okay, dia tidak expressed. Kita akan tengok lebih tu lagi lah. So the reason for this existence of the imprinting genes on cerebral chromosome lah. So common 15 is one of it, and there are genes that are only expressed from either a maternal or a paternal chromosome, and there are probably about 100 such loci altogether. So examples supporting this principle is the insulin-like growth factor IGF-2 system. So it has been shown that the imprints are set up during gametogenesis. So the whole embryo clones met the nuclei from the primordial germ cell show an erasure of the origin imprint from E1115. E1115 ialah masa dia dalam kandungan tikus. Masa dia mengandung dalam day ke 1115 tu. 11.5 itu. So probably associated with the genome-wide demethylations of the DNA of this cell. Okay? Gene itu dia kalau dia tak expresskan, it will be methylated. Faham tak? Okay, class. Listen very carefully. During the fusions of nucleus from a sperm and nucleus from the ovum, there's a fusions kan, the restorations of the genome kan, from N and N become 2N, betul tak?
Yes, doktor.
Yes.
Yes.
Remember this kan? You remember kan?
Remember?
Yeah.
Okay. Now, at the very beginning of life, saya pernah cakap bahawa the zygotic expression dia tak muncul lagi tak? Zygote tu belum buat protein dia sendiri kan? Kerana dia depend on the maternal contribution, betul tak?
Betul.
Yeah.
Ya. Jadi apa yang berlaku pada jin-jin yang terkandung dalam zygote itu? Kenapa dia tak express?
Because it's not needed.
It's not needed. Kenapa dia? Because all of this gene dia kena methylated. Dia ada methyl group yang kena remove, baru gene itu akan express. They are not being methylated. Only by removing dia punya methyl group tu, baru gene itu akan mula expressed. Okay?
Clear, doktor.
Okay, so the zygote period ataupun the time point when the zygote start to express or making their own protein is called zygotic transmission. Remember this word? Pernah saya dengar ini? Zygotic transmission? Tak pernah dengar? Saya tak pernah cerita ni. Saya rasa pernah cerita pada first lecture kan? Ataupun you tak ada bayangan lagi? Okay, never mind. Contohnya ya, contohnya in zebrafish. The first 3 hour or first 3.3 hour to be exact, all the expressions is all from the mother, from the maternal. Okay? Whereby after 3.3 hour post-fertilization, then the zygotic transmissions start. That means the zygote make their own protein, or the transcription of the genes from the zygotic starts. Faham tak?
Faham, doktor.
Ya, tapi sebelum itu semuanya proses pada awal tu adalah daripada contribution from the maternal. Betul?
Betul.
Betul, doktor.
That's why kalau kita tengok contohnya, kita ambil contohnya ikan kan, telur ikan besar tak? Kita nampak kan telur ikan tu kan?
Ya,
Ya. And then sperm tu tak nampak, betul tak?
betul.
Ya. Kenapa telur tu kena besar? Kerana dia kena store banyak information. Antaranya selain daripada the nutrients for the early developments, but at the same time dia pun carry banyak proteins yang diperlukan untuk pembahagian pada peringkat awal. Sebelum zygote mampu buat protein dia sendiri, synthesize their own protein. I hope this is clear enough. Huh? Boleh tak?
Boleh, doktor. Clear.
Boleh, doktor.
Okay, good. Okay, so kalau kita tengok X inactivations, for all female mammals, only one copy of X chromosome become heterochromatic, and inactive sense is visible in the bar body within the nucleus. So this occur at random at the primitive stage, such as either the maternal-derived or the paternal-derived X become inactive. So the inactivations of one copy of the X chromosome produce a mosaic with respect to any heterozygous locus. So X inactivation depend on the presence of the chromosome on X inactivations center. Okay, so this contains a gene called the exist, which is pronounced an exit, which encodes a non-translated RNA. So exit is critically important for X activations and work only on its own chromosome. Contohnya kalau kita tengok di sini. Ya, kita ada exit di sini. So ada blocking factor akan blockkan dia and remove the blocking factor, and this exit become more active. Okay, so bila dia active, then the RNA will cut for the chromosome. So the inactivated stage is reinforced by the histone acylations, macro H2A recruitments, and the DNA methylation. Okay. Now, bagaimana kita screen for mutant? Bagaimana kita boleh buat mutant? Okay, either we do the forward genetics or the reverse genetics. So many interesting mutants have arisen spontaneously, but even more has been recovered in the large-scale screen of the Drosophila, the C. elegans, and zebrafish in mouse. Okay, especially zebrafish in late 80s or beginning of the 90s, they screen a lot of this zebrafish. Okay, but the principle is simple and relies just on the basic Mendelian genetic. Contohnya, this is ini nampak tak ini male kan?
Yes.
Right?
Yeah.
This is a male fish and this is a female fish. Yeah. So mutagenesis, that means we expose this male fish with the mutagen. Okay.
Okay.
Alright, mutagen. Kita harap bila sperm dalam sperm itu dia punya genetik boleh mengalami certain alteration lah. Okay. Kita supaya nanti mutant boleh dihasilkan. So we cross dia dengan wild type. Okay, this is a wild type female. Kita cross dia kan, dan kita dapati di sini untuk family 1 dia normal semua. Okay, untuk yang di sini the family kedua ni some ada mutants. Kita nampak. Bagaimana kita nampak? Biasanya kita nampak dari segi dia punya pigment. Because this fish, zebrafish, dia ada 3 pigment yang utama, iaitu kita ada melanophos yang encode pigment hitam, kita ada xanthophos yang encode pada pigment warna yellow, dan kita ada iridophos yang encode pada silver. silver punya pigment. So daripada the color code itu kita boleh nampak. Ya, kita boleh nampak. And kita cross untuk family ketiga all normal. And di sini kita ada some mutants. Okay, so kita screen for the mutant, nanti ialah kita crosskan dia lagi. Okay, untuk yang ini F1 individual, kita ini F2 family, dan kita ini F3. Dan kita boleh cross dia lagi. Okay, kita cross dia dan kita harap contohnya di sini, kalau kita cross dia, the possible F2 crosses kan, daripada sini, F2 punya family kita crosskan antara F2 ini dan 75 yang normal dan 25 yang crosses ni akan menghasilkan mutants. So maksudnya 3 kepada 1 ikut Mendelian. Masih ingat tak Mendelian punya ratio 3:1 kan?
Yes, doktor.
So
Yep.
so 35, this one is 75, 25, so 3:1 lah. Betul tak dia punya ratio?
Yeah.
Ya? Okay. Dan kalau kita buat positional cloning, kita boleh buat juga positional cloning. Okay, kita boleh gunakan macrosatellite punya marker tu dan kita di sini marker allele ABC, di sini ialah G is negatif ni maksudnya kita dah keluarkan. Ya, kita keluarkan dan kita kawinkan dia, kita matkan dia dengan yang normal itu dan kita dapati di sini ialah kita ada A besar, A kecil, B besar, B kecil, C besar, C kecil. Dan lepas tu kita boleh check dia punya genotype dia di sini. Ya, so di sini kita dapati ada yang sudah hilangkan di sini. Ya,
Yeah.
ada missing point dia di sini kan? Certain missing. That's mean the gene already been knocked out at certain position. Okay? And this is the observable mutants mutant yang dia contohnya di sini dah hilang semua. Ya? Nampak di sini? Kalau kita tengok di sini dia punya profile dengan sini lain kan?
Yeah.
Maksudnya kalau dia sini tak ada maksudnya gene itu telah pun di knock out, betul tak?
Yeah.
Okay? Satu lagi ialah transgenesis. Transgenics is for the case that extra genes has been introduced into the genome. Maksudnya kita knock in. Kita buat extra, kita buat extra gene tu kepada certain organism, kita nak tengok apa yang akan berlaku kepada organism itu. And knockout is which a gene has been removed from the genome. Okay, so in mouse and zebrafish, the genes is introduced by injecting DNA right into the fertilized eggs. I have done a lot of this lah, kita buat transgenic ini. Ya, just do injection saja the DNA, then we alter the DNA. So in Xenopus ialah katak. The DNA is incorporated into the sperm, and in Drosophila and C. elegans, the transgene is incorporated into the transposable elements which is injected into the germlines of the embryo. So each model animal, they will use different techniques. Okay, kita tak boleh apply the same technique into all animals. Okay, and the transgenes are usually designed so that the expression is regulated by a promoter within the insert. So kita insert certain tu, kita ada promoter and we can check whether the genes is expressed or not. Or we also can have the enhancer trap. Okay, the enhancer trap consists of the reporter genes such as the LEXZ genes coupled with a minimal promoter that can bind the transcription complex within the range of an enhancer. Kita trapkan enhancer tu, bila kita trap enhancer tu, enhancer tidak express. You know, for a gene to express, enhancer tu kena berfungsi kan? Ya, kita ada promoter, kita ada enhancer. The enhancer is the one that enhance the promotion of the promoter. Betul tak? It could be upstream of the promoter. Masih ingat tak?
Ingatlah, doktor.
Okay, good. And targeted mutagenesis is that the introduction of the specifically engineered mutations at the desired site in the genome is a technology that has been brought to the high level of the mouse, but it's not generally available for other species lah. Kebanyakannya kita buat ini untuk tikus. Karena tikus ialah mammal, di mana dia paling dekat dengan manusia lah. That's why kita gunakan mutagenesis ni targeted ni untuk tipus. Okay, so it has mainly been used to produce knockout or loss of function mutations in particular genes selected because they were thought likely to be of the developmental or medical importance yang hanya ada medical ataupun developmental punya importance tu baru kita kita select gene itu. And the targeted mutagenesis depend on the homologous recombinance, which is in the direct replacement of the genes of the modified versions. So example, in the mouse, they are embryonic stem cell that can be engineered and then incorporated into the mouse to produce a germline chimeras from which the new strain can be bred. So I believe Dr. Chung will teach you how to generate a mutant mouse, right? Because you are taking animal biotechnology, right?
Yes.
Yes, doctor.
Yeah.
Yes.
And basically, it's very easy actually. In theory, it's very easy, but come to practical, it might be very challenging lah. Everything on paper sangat senang kan. In theory, kita cakap saja mah. But once you do the hands-on, then you know. Okay. And other methods of producing specific inhibitions of the knockout, contohnya dominant negative reagent. Kita boleh gunakan domain swap method, antisense like RNNI or morpholinose. Like me, I use a lot of morpholinose to knock out the gene. We have the specific neutralizing, the antibody pun boleh gunakan juga. Dan kalau kamu nak baca dengan lebih, then you go to YouTube lah. These are two YouTube movie that you may watch to look how the inhibitions of the gene can be done. Okay. And lastly is the gene duplication. So gene duplication is probably a major source of the evolutionary novelty. Not all organisms, they go through gene duplications, yeah. And we found that gene duplications happen a lot in fish. So that means one copy could continue to be the functional gene, where other could be accumulate the mutation, such as that is required for a normal or advantageous functions. But no people know why genes is duplicated or the genome being duplicated. No scientists know it for sure. They only can postulate, but because this happened million years ago, so they can also postulate. But no people really know why they are being duplicated. And the alternative of the second copy could accumulate the deleterious mutation until it become non-functional or maybe eventually not even expressed, which become a pseudo gene. Pseudo mean palsu, gene yang palsu. Dia memang, it look very similar or exactly the same like the gene, but they don't have the function. They become the pseudo gene in our genome. So the extreme case of the gene duplication occur when the entire tetraploidization genome become duplicated with a double of the chromosome number. So 2 tetraploidizations even may occur at the time of the regions of the vertebrates. Okay. Please mute yourself, yeah. Kuratu Aini, can you please mute yourself?
Kejap. Adik. Aku tekan apa? Sebut nama kau.
Okay, so the 2 tetraploidizations event may have occurred at the same time of the origins of the vertebrate and temporarily boosting the gene number from contohnya from 20,000 to become to about 80,000. So tetra, that's been the time 4, so from 20,000 to 40,000. And we have the pseudo genes, the pseudo alleles as well. They look like allele, but generally have the similar expression pattern of the function, but they are not allele because they are occupy the distant genetic loci. Okay, so this is a overview for you to understand the developmental genetics. Okay, of course, actually there are more. You want to read more because we cannot tell you everything. So it will be good if you can have a read on this book, okay, from Jonathan Slack, to understand more about the developmental genetics. Actually, this is a very important topics for you to understand. Okay, how genetics how developmental genetics can play around in the field of developmental, okay, in developmental biology. So far okay. Getting tougher kan?
Yeah,
Yes, doctor.
Yeah.
Yes,
difficult to
doctor.
digest.
I can understand because honestly, if I ask other lecturers to teach, they also feel very difficult, very challenging, because if you are not from the field of developmental biology, it's not easy for you to understand. I can understand that because the content that you are using here is the exact same that I use in Bath University. Actually, a lot more, but we try to simplify it. If not, you akan pengsan. Contohnya kita slide di dekat overseas mungkin ada 60-70 slide. But I try to consolidate dia supaya dia lebih senang untuk kamu faham. Saya kadang-kadang pun dapati not easy to explain because you all punya asas tidak cukup. Bila asas tidak cukup, susah saya nak explain supaya kamu boleh faham. But I will try my very best, being a developmental biologist, to make you understand in a easier way, supaya you faham apa yang saya cakap. I don't expect you 100% understand this, but at least 80 to 90% you understand my lecture. Boleh?
Boleh, Doktor.
Boleh,
Doktor.
Boleh doktor.
Okay. So it will be good. I'm going to like to have all the lecture. I'll make it visible because most of my lecture I hidden it from you. So just now I forgot to make it visible. So you can go to Ellipse. If you see no notes there, you just remind me to make it visible to you because all the material already there. Okay.
Okay, Doctor.
And so it will be good.
Doctor.
You read the notes before you attend my class. That will be better so that you can understand easier. Okay, you can master the subject in an easier way if you have done the pre-reading beforehand. Okay?
Okay, doctor.
I understand. Okay, all the best for you and take good care.